Enterococci also constitute a large preparation of autochthonous bacteria associated with mammalian gastrointestinal tract. The resistance of enterococci to pasteurization temperature and their adaptability to different substrate and growth condition in food product manufactured from raw material and heat treated food product is of the great significance. Enterococci may constitute an important part of the microflora of the fermented cheese and meat. E. coli strain are associated with foodborne gastroenteritis. These are specifically gram-negative aspergillus rod that ferment lactose and produce dark colonies with metallic sheen on endoagar. This organism grows well on large number of media and many foods. They grow over wide range of temperature from 4 to 46 degrees Celsius and pH from 4.4 to 9.0. However, they grow very slowly in food held at refrigerated temperature less than 5 degrees Celsius. They belongs to a family of Enterobacteriaceae, and the organism is also an indicator of fecal pollution. The organism is also capable of producing acid and gas and off flavor in food. E. coli strain involved in foodborne illness can be placed into 5 groups. Number 1, enteropathogenic EPEC. Number 2, enterotoxigenic ETEC. Number 3, enteroinvasive EIEC. Number 4, enterohemorrhagic EHEC. And 5, facultative enteropathogenic EFEC. This organism also grows in presence of bile salt. The primary habitat of E. coli is of the intestinal tract of most warm-blooded animals. E. coli 0157.H7 strain are unusually tolerant of acidic environment. Lactococcus is a genus of lactic acid bacteria that were formerly included in the genus of Streptococcus group N, which is called group N Streptococci. They are also known as homofermenters, meaning that they produce a single product of glucose fermentation. They are gram-positive, catalase negative, non-motile coccus that are found singly, in pairs or in chain. Some of the strain of Lactococci are known to grow or below even 7 degrees Celsius. Lactococci are intimately associated with dairy products. These organisms are commonly used in the dairy industry in the manufacture of fermented dairy product like cheese. They can also be used in single strain starter culture or in mixed strain culture with other lactic acid bacteria such as Lactobacillus and Streptococcus. Their main purpose in dairy production is the rapid acidification of milk. This causes drop in pH of the fermented product, which prevent the growth of spoilage and other pathogenic bacteria. This bacteria also plays a role in flavor of the final product. Dairy lactococci have also been exploited for several industrial fermenters in the biotechnological industries. They are easily grown at industrial scale on cheap whey-based media. Lactococcus lactis, subspecies lactis include species formerly designated as Streptococcus lactis, subspecies lactis. Lactobacillus lactis subspecies cremoris is distinguished from Lactobacillus lactis subspecies by the inability to grow at 40 degrees Celsius and grow in 4% NaCl and hydrolyze arginine and ferment ribose. Lactobacillus acidophilus, Lactobacillus bulgaricus, Lactobacillus helveticus, Lactobacillus plantarum, Lactobacillus casei, Lactobacillus lactis and Lactobacillus fermentum belonging to the important genus that are all rods, usually long and slender, and in some of the species form chain. Lactobacillus are aerotolerant or microaerophilic, but some ferment sugar chiefly to lactic acid if they are homofermentative. The heterofermentative species beside lactic acid also produce small amount of acetic acid, carbon dioxide and trace amount of volatile compound such as acetaldehyde and alcohol. The homofermentative species like Lactobacillus include Lactobacillus bulgaricus, Lactobacillus casei, Lactobacillus helveticus, Lactobacillus lactis, Lactobacillus acidophilus grow optimally at 37 degrees Celsius. Lactobacillus fermentum, Lactobacillus brevis are typically example of the heterofermentative Lactobacillus group and grow very well at high temperature. Lactobacilli are of considerable importance in the food industry as they ferment sugar to lactic acid and other desirable flavoring compound and thus used in the production of fermented plant, dairy and meat product. However, they are also implicated in the spoilage of wine and beer. The organism normally occurs on plant surface silage, manure and dairy product. They are quite fastidious in their nutritional requirement as they are unable to synthesize certain vitamins and they require therefore media need to be supplemented with these vitamins for their growth. Some of the strains are psychotropic in nature and are thus involved in the spoilage of refrigerated meat. On the other hand, thermoduric property like resistance to pasteurization temperature of some of the thermophilic strain of lactobacilli are quite useful in the manufacture of certain variety of cheese like Swiss cheese. Some strain of lactobacilli also show probiotic attributes and are finding application in functional probiotic foods as well as in pharmaceutical preparation. Leuconostoc is a genus of gram-negative bacteria placed with the family of Leuconostocia. They are generally ovoid cocci, oven-forming chain. Leuconostoc species are intrinsically resistant to vancomycin and are catalase negative, which distinguish them from staphylococci. All species within the genus are catalase negative, homofermentative and are able to produce dextran from sucrose. They are generally slime forming, blamed for causing the stink when creating a short dough starter. Some species are also capable of causing human infection. Leuconostoc species along with other lactic acid bacteria such as Pediococcus and Lactobacillus species is responsible for the fermentation of cabbage to sauerkraut. In this process, sugar in fresh cabbage are transformed to lactic acid, which give it a sour flavor and good keeping qualities. Pseudomonas fluorescens is a common gram-negative rod-shaped motile bacterium. This organism is psychotropic in nature and grows at refrigeration temperature below 7 degrees Celsius. It has an extremely versatile metabolism and can be found in soil and water. It is an obligate aerobe, but certain strains are capable of using nitrate instead of oxygen as a special final electron acceptor during the cellular respiration. Optimal temperature for growth is lying between 25 to 30 degrees Celsius. It tests positive for the oxidase. Pseudomonas fluorescens also a non-saccharolytic organism. Heat-stable lipase protease are produced by this organism, and these enzymes cause milk to spoil by causing bitterness, casein breakdown, ropiness due to the production of slime and coagulation of protein. Pseudomonas aeruginosa is a gram-negative aerobic rod-shaped bacterium with unipolar motility. An opportunistic human pathogen is also an opportunistic pathogen for plant. Pseudomonas aeruginosa is a type of species of the genus Pseudomonas. Gram-stained bacteria, pink red rod, secretes a variety of pigment including pyocyanin, which is blue-green, pyoverdine, which is yellow-green as well as fluorescent, and pyorubin, which is red-brown. Pseudomonas aeruginosa is often preliminarily identified by its fluorescence and grape-like or tortilla-like odor in vitro. Definitive clinical identification of Pseudomonas aeruginosa often includes identifying the production of pyocyanin and fluorescein, as well as its ability to grow at even 42 degrees Celsius. Pseudomonas aeruginosa is also capable of growth in diesel and jet fuel, where it is known as hydrocarbon using microorganism or HUM bark, causing microbial corrosion. Pseudomonas aeruginosa is also considered by many as a facultative anaerobe.